Hi everyone, my name is Madison Goforth. I am an undergraduate student worker in Dr. Carrie Cooper's lab. And for my project, I would like to discuss at the Food Safety Consortium is microbial diversity of different melon types grown in different regions in the United States. So a little bit about the introduction and some background on microbial diversity. There's only been one study that has been published on the microbial diversity of melons. However, if we were to take a look at this diversity, it's only on one type of melon, cantaloupe. And for the study, they did more of a field study where they swabbed hands of workers, of equipment, and processing in the field instead of looking at different locations. This was a great study, though, to publish and have knowledge on microbial diversity. So why this is important is because majority of our melons are grown in California and Arizona. So really taking the time to look at microbial diversity on melon rinds can help us open up new avenues, such as identifying potential protagonistic and antagonistic bacteria that can be used against foodborne pathogens like Listeria or Salmonella that are really prevalent in these melon outbreaks. This study also lays down a foundation for future research on melon varieties and their regions that can contribute to areas of food safety, plant health, and improving shelf life. The aim of the study was to determine variation of microbial diversity between types of melons, as well as the various regions that they are grown around. Some of our methods employed were sampling from 7 locations, doing extractions of these swabbing of the rinds, and then sequencing with Illumina MiSeq and using CHIME analysis to identify alpha and beta diversity. So for some results, for our figure 1, we have relative abundance of the top 13 bacterial families associated between all 7 regions of 4 melon types. Some of the big contenders are Enterobacteriaceae, Microbacteriaceae, and a couple others that were really prevalent. In figure 2, we see the Shannon diversity, which is abundance and evenness of our bacteria, and significance was found primarily in type of melon. In our figure 3, we also show the operational taxonomic units, which are basically the number of different bacteria between each subsample. And as you can see, for statistic variance, it was high in type of melon as well. In our figure 4, we have the Bray-Curtis PCOA, which is a beta diversity, and it shows clustering of bacterial communities by region. So Arizona being red and California being blue, and so on and so forth. For our tables, we're just showing the total number of melons sampled across the 7 states. For our conclusions, looking at alpha diversity, they showed a great significant difference in bacterial diversity based on type of melon, while beta diversity analysis demonstrated that melon samples clustered with similar bacterial communities is more based on location of growth compared to type of melon. So for some future directions, we can use this information to identify protagonistic and antagonistic bacteria that can work against foodborne pathogens like Listeria and Salmonella. This is important, especially for our beta diversity between different regions, because there are some that are prevalent in areas of California and Arizona, since that's where we get most of our melons. It would also be useful to identify variation in spoilage bacteria, as well as further identification of bacterial diversity of melons and regions. I'd like to acknowledge and thank the Cooperative Extension's faculty between all 7 universities involved in this project. Thank you very much.